a component of a biomolecule, proteins, amino acids, the building blocks of proteins. We'll look at the peptide bond and also the amino acid reactions. I don't have an outline slide for this particular section, so I'm just going to state some of the things that I hope you can answer, or some of the types of questions I hope you can answer when we're done here. You'll be able to, or should be able to, draw the structures and know the properties of amino acids, particularly the acid-base properties of amino acids. We're going to look at the reactions that amino acids undergo, take a look at stereochemistry and optical properties. Also, we at some point are going to look at how amino acid mixtures are separated, and analyzed. Also, the fundamental structure of proteins. So let's start by taking a look at a general or a typical amino acid. Every amino acid has an alpha carbon. And at physiological pH, the amino group is protonated and the carboxyl group is deprotonated. So when I say physiological pH, I mean a pH of 7.4. So physiological pH is a little bit basic, not 7. Well, we have also pKa's or PKs for ionizable side chains, and also the amino and carboxyl groups, which you will also learn to know. The pKa, and I'm just going to say it here, and remember, the pKa is the negative log of the K sub A, or that association or dissociation constant that we talked about when we looked at the Henderson-Hasselbalch equation. So, the general structure, we have that alpha carbon, we have the amino group, and we have the carboxyl group, and then we have a hydrogen, and then we have some R group or side chain. In the simplest amino acid, this is a hydrogen atom. So, and that would be glycine, and we'll take a look at all these individually. Glycine then is considered a non-chiral molecule. Remembering back to your OChem and chirality, a chiral molecule has a carbon bonded to 4 different groups. And in glycine, we have 2 hydrogens. All the other amino acids are going to be chiral molecules, because this R chain will be different. It will not be a hydrogen group. So there are several ways to show the structure of an amino acid. This is one, obviously. This is a ball and stick model. It shows, if you had a model kit and you put this together, it would show the tetrahedral structure, which is actually depicted here. So they are tetrahedral. Bond angles, remember, for tetrahedral structures are 109.5 around this central carbon. So classifying amino acids, there are 9 polar amino acids. Non-polar amino acids. Jumping ahead again, I like those polar ones. Anyway, non-polar amino acids then are going to have hydrophobic properties. So when they are found in a protein, they're going to be found on the interior of that protein, because that protein, again, is in a solution that is water-based, or an aqueous solution. So these amino acids are going to be on the interior of a protein, and actually are responsible for driving protein folding. So what do we have? We have leucine, and we'll go over this again later, but in addition to the name and the structure, you need to also know the 3-letter abbreviation and the single letter abbreviation for each amino acid. And if there is an associated pKa, you will have to know that as well. So nonpolar or hydrophobic amino acids will not have pKas because they have nonpolar side chains. So we have leucine, and actually we're going to draw all of these together, so I'm not going to spend a lot of time. But here is the alkyl group, or the R group, for leucine. We have proline. Now notice that proline is a little different. It is the only amino acid that has a bond between the side chain and that amino group. So alpha carbon, our carboxyl group, our amino group, and here's our R side chain, which wraps around and links with that amino group. Alanine, step up from glycine, and the simplest next to glycine just has a methyl group attached as its R chain, and then we have valine as well. Other nonpolar or hydrophobic amino acids, methionine. And we'll find that that plays a special role in a couple of different ways with respect to proteins and also as side chains. Here is that sulfur that I mentioned early on, I think in the first video. Methionine contains a sulfur group. Tryptophan. This is based on, this is aromatic. We have that benzene ring, and this is its, well actually I guess, let me take this back. Here is our carbonyl, our carboxyl group, our amino group, and the side chain for tryptophan. Phenylalanine, also aromatic with that benzene ring and its as part of its side chain, and isoleucine. Polar amino acids. So glycine, depending upon the text you're looking at, may be classified as polar or nonpolar. At physiological pH, it is basically, this group is non-protonated, and actually these are not shown at physiological pH because this would not be protonated. So at physiological pH, this is deprotonated, this is protonated, so we have a cancellation of charge here. But glycine, I probably won't ask you to classify glycine because of that. Serine has that hydroxyl group. Remember that that oxygen is more electronegative than the other molecules in the, or atoms in the side chain, so it pulls the charge toward that oxygen, so we have a partial negative here. Same in asparagine. We have lone pair on nitrogen, and we have two lone pairs on this oxygen, and glutamine, same case. You'll see the only difference here between asparagine and glutamine, and I hope you'll, when you're learning your amino acids, kind of think about these things in the learning process. We have only one carbon between our alpha carbon and our carbonyl carbon, and here we have two carbons. Threonine, also considered a polar molecule. We have that hydroxyl group. Cysteine, here's that SH group again. This SH is going to allow it to react with other cysteine molecules, and we'll take a look at that reaction as well. Tyrosine is another aromatic but also polar amino acid. Again, here's that benzene ring, but we have a hydroxyl group here that makes that a polar molecule. And histidine. Histidine is Actually, I think we'll just come back to that. You might notice that we have this nitrogen that is partially protonated. When we draw these together, I'll discuss more what happens to this. It does have a pKa of about 6, so at physiological pH, this is only about 10% protonated. But as I said, we'll come back to that when we draw these amino acids together. There's some amino acids that are acidic. We have 2. So these are both carboxylic acids, or they're acidic because of their carboxyl groups in their R chain here, which has an ionizable hydrogen, and that this will be deprotonated at physiological pH. And again, you'll notice in comparison, like we saw between asparagine and glutamine, that there is 1 carbon in between the alpha carbon and the carboxyl group, and there are 2 between that alpha carbon and the carboxyl group here. 2 basic amino acids, lysine and arginine, and these are basic because of those nitrogen atoms that are in the side chain. We'll look at the pK definitely for lysine. I'm not sure that we take a look at it for arginine, but again, when we draw these, I'll give you all the pKs that you are required to know. So, these are the ionic forms of amino acids shown without their side chains. So if they had ionizable side chains, we would have to take that into consideration. Excuse me. But right now we just want to look at what the ionization state is at different pHs. So at pH 7, net charge of this amino acid, which we're going to say it has a nonpolar or non-ionizable side chain, because that carboxyl group is deprotonated and the amino group is still protonated. At a lower pH, and we'll look at this again in detail, we'll draw some of these with some titration curves with real amino acids, not just these general forms. Both the amino nitrogen and the carboxyl group are protonated essentially. So because this is not charged and this is, we have a net charge of 1. Okay. If we go in the other direction, if we get up into a higher pH where the pK for the amino group is, then we are going to deprotonate that nitrogen, and now we have no charge on the nitrogen and we have a negative charge on our carboxyl group. Thus, the net charge here is -1. I'll repeat this again at some point, but the pKa for the the ionizable hydrogen here is 2.2. The pKa for the ionizable hydrogen on this amino group is 9.4. Finally, we're going to look at the formation of a peptide bond, and we'll also walk through this reaction together. So here we have the amino group, the carboxyl group on one amino acid, amino group on another amino acid. So we have 2 different amino acids here. Don't know what they are. Well, we'll just pretend these represent the R groups. And what happens is we are going to lose an oxygen from this amino acid, and we're going to take 2 hydrogens from this one, which releases a molecule of water and forms that peptide bond. So we're going to form a COO to NH bond when we form that peptide bond. So this is another way to look at this, maybe a little easier, except it would be easier if these hydrogens were shown as individual bonding. But we're going to remove this oxygen, two of these hydrogens, release that water molecule. So this is an elimination reaction or dehydration reaction. And we get that double bond, C double bond O is maintained here. We get the C end bond and a bond to this H. So what we formed here is a dipeptide. So we have R1 and R2 indicating that we have two different types of amino acids. This figure is, and I'll try to show this also when we're drawing this, is trying to depict the coplanar relationship between this carbon, nitrogen, oxygen. Remember, this is a double bond here, and what happens is there is a flipping. So we've got movement of electrons between this bonding region and this bonding region, which makes this structure very rigid and all in one plane. So there's no rotation between this carbon and nitrogen, as you would expect with a single bond. It is going to remain rigid.